In this presentation, I'll try to convey an understanding of how we know what we know about the brain. This relates back to our previous discussion about maintaining scientific skepticism. I'll talk a little bit about the history of brain research and then some of the more modern techniques for studying brain function. I'd first like to thank and acknowledge the APA for putting together a wonderful guide for this unit, more of which is available to teachers at apa.org. It's important to understand the role that technology has played in helping us study brain function. At first, as you can imagine, there was little technology available. This, of course, did not stop people from asking questions and posing answers. At first, most of what was known was based on observation of both normal function and dysfunction due to illness and damage. Over time, three systems or methods emerged for studying brain function. First is through brain lesion or ablation, which is the removal of brain tissue. Second is through stimulation of brain regions. And the third is through imaging of the brain by various methods. So if we go back through recorded history, we see that several people made some very astute observations about the brain, even in the absence of testing methods or technology. In ancient Greece, Hippocrates argued that the brain was the center of emotion and thought. This was contrary to other views that persisted for centuries that argued for the heart or even the liver as the center of emotion and thought. Gall, a Roman of Greek descent who was living in Turkey at the time, claimed that fluids in the brain, particularly in the ventricles of the brain, were responsible for sensations, memory, movement, and other functions. Ibn al-Haytham, also known as al-Hasan, a Muslim scholar in Cairo, produced a stunningly accurate theory of vision in which he argued for light rays entering the eye as the cause of visual perception. This was contrary to two other leading theories that were popular at the time, one by Ptolemy and one by Aristotle. So people have been speculating and arguing about the role of the brain and the nervous system for a very long time. Sometimes this argument has led to false conclusions that nevertheless gained considerable attention. Phrenology was just such a case. Franz Gall and Johann Spurzhein claimed that the contours of the skull reflected personality traits and moral character. Of course, when subjected to further scrutiny, the theory falls apart very quickly. One of the most influential cases in the study of the brain is Phineus Gage. Gage was, by all accounts, a pretty level-headed guy, relatively successful at his job, and generally normal. After his accident, in which a tamping iron was blown through his skull, his personality changed dramatically. Now, certainly the trauma of having a 3-foot iron bar driven through your head would have an effect on anyone. But in the case of Phineus Gage, the resulting brain damage fundamentally altered his behavior. From this case, we've learned the role of the frontal lobes in moderating and controlling emotions. Around the same time, actually about a year after Gage died, a French physician by the name of Paul Broca conducted an autopsy on a patient known as Tan. Tan had apparently lost the capacity to produce language. The autopsy showed damage to the left frontal lobe, leading Broca to hypothesize that this specific region of the brain was responsible for speech production. Later evidence has corroborated this claim. Another physician, a German this time, Karl Wernicke, discovered that damage to the left temporal lobe resulted in the loss of language comprehension. These two findings were very important in that they demonstrated the localization of function in the brain. They also showed that functions such as language are very complex and utilize multiple areas of the brain. In all of these cases, we've learned a great deal about the brain through brain injury. Another method of identifying brain function is through ablation, or the removal of brain tissue. So obviously we can't just go around taking out parts of people's brains and to see what happened, but we can perform procedures on non-human animals. For example, we know that removing a portion of a rat's hypothalamus can lead to the rat either losing all desire to eat or to the rat continuing to eat unabated, depending on which part of the hypothalamus is removed. Removing and then staining the neural tissue with a silver nitrate solution has also allowed scientists to observe the structures of individual neurons. This system, which was discovered and perfected by Gall and Golgi, led to a breakthrough in brain research. A better understanding of the nervous system and the structure of the neurons in particular led to research by Hess and Penfield in electrostimulation. So by stimulating different cortical areas, such as the back of the frontal cortex, at particular sites, caused movement of different body parts, enabling mapping of the motor cortex. Walter Hess inserted electrodes more deeply into the brain of non-human animals that were under anesthesia. After the animals recovered from the surgery, he was able to relate start-stop functions with specific brain structures. For example, once again, the start eating and stop eating functions associated with areas of the hypothalamus. Further advances in technology led to better imaging systems. Perhaps the first was the EEG, or electroencephalogram, which measures the electrical activity of the brain and records it or reports it as brain waves on a printed surface. A lot of what we know about sleep patterns is a direct result of using EEGs to measure brain activity. A CAT scan, or CT scan, is a computerized axial tomography of the brain, or the region being studied. The CAT scan uses X-rays to show a two-dimensional slice of the brain. CAT scans are very effective at finding and showing tumors and brain damage. MRI, or magnetic resonance imaging, also uses a very large magnet to image the brain. This produces a more detailed image than the CAT scan, but it's limited in its use. For example, patients with pacemakers or metallic implants of any kind cannot undergo MRI. fMRI, or functional magnetic resonance imaging, allows for a real-time look at brain functions. And this has been very effective in observing the brain as it works to complete tasks such as reading, or writing, or playing chess, even exercising. PET scans are also very effective in seeing the brain in action, but are also limited in use. The PET scan requires the injection of a mild radioactive substance, and although it is a low amount of radioactivity, multiple uses of PET scans are discouraged and therefore limited. So the advent of more and more powerful computers has allowed a breakthrough in brain imaging. Today there's multiple varieties of brain imaging techniques. Beam systems utilize input from multiple EEGs to create a highly specific map of brain activity. An MEG utilizes magnetic fields to detect neural activity that is too brief to be picked up by a PET scan or an MRI. Scientists can now also use a TIMS device to temporarily depress or enhance brain activity in a specific area, effectively allowing researchers to turn on and off certain functions of the brain. Breakthroughs in genetics have also allowed scientists to remove specific genes from mice and other organisms to help them understand the link between genes and behavior. For example, mice that are lacking one of the genes responsible for regulating the neurotransmitter dopamine act as if they are permanently high on cocaine. Optogenetics also allows neuroscientists to insert genes into neurons of non-human animals that cause the neurons to become sensitive to and excited by light. Shining a light on that part of the brain will then activate those neurons, and this allows specific activation of only the modified neurons, allowing their functions to be better understood. So we can now utilize a variety of methods for understanding the brain. Of course, the technology itself is only a means to an end. Proper application of the scientific method and adherence to strict scientific and ethical guidelines is the only way to responsibly research and uncover the secrets of the brain. So you can now go on and view the video on Phineus Gage and answer the journal questions, and then please be sure to take the quiz on both this lecture and Phineus Gage.